continue discussions of clinical chemistry. But before I start our lecture, actually, I have explained something wrong that is related here. Actually, that is correct. It's completely correct that the ALT that is present in the liver only, so it's more specific than the AST. So ALT is present in the liver, okay, mainly, but the AST that can maybe present in the liver, in the heart, and the muscles, and it is present actually 80% of it is present in the mitochondria. So that's what I want just to revise it. And again, if the AST more the ALT, that's mean it's severe damage of the liver, but actually ALT indicate more the liver damage because it related to the liver only. But if AST more than ALT, that's mean alcoholic and acute liver failure. If there is ALT, that's mean it's a viral hepatitis. So that is correct completely. ALT present in the liver only, AST present in the liver, in the heart, and also in the muscle. Now we go to our lecture, which is here. We discussed also that before is here the heme, and that actually happened inside the spleen and the macrophages. We have the heme plus oxygen that is led by the heme oxidase enzyme production of the biliverdin plus CO plus iron, and then the biliverdin with the NADPH positive by biliverdin reductase enzyme, then it lead to the bilirubin plus NAD positive, and then the bilirubin with the oxygen that lead to the oxidized bilirubin. Type 2 of the Greigel-Nijar syndrome, type 2 because it has type 1 and type 2. Type 1 is more severe because of the absence of uridine diphosphate glyceronyl transferase enzyme, and we sometimes said 1A1. It's decreased, but type 1 is absence, so it's more severe, okay, and it cannot be treated. Again, ketone bodies is acetoacetate, acetone, beta hydroxybutyrate, and I explained in details how it forms these ketone bodies. Also alkaptonuria, also I explained it in more details. So we have here alkaptonuria, and that is due to the mutation on the HGD gene, which is known as the homogentisate 1,2 dioxygenase enzyme. So what we called it as homogentisate. H is homo, G is gentisate, D dioxygenase, and we have to say the number. So homogentisate gentisate 1,2 dioxygenase enzyme. So alkaptonuria, it's due to the mutation in HGD gene, which codes the enzyme homogentisate 1,2 dioxygenase enzyme. So to understand how that is happen, we have to know all the cycle that is lead to this, and I explained it in a very detailed form. So as you can see here, we have the phenylalanine, which is amino acid, and we described also there is a playlist of all the amino acids, so you can return back what is the phenylalanine. And then the phenylalanine by the phenylalanine hydroxylase enzyme, that it will be turned to the tyrosine, and then the tyrosine transferase enzyme that is lead to the 4-OH phenylpyruvate, and then by HBD dehydrogenase enzyme that is lead to the homogentic acid, and the homogentic acid by the enzyme that we called it as homogentisate 1,2 deoxidase enzyme or deoxygenase enzyme, because it's so important to produce here is correctly, that is lead to the production of the mild acetoacetate, as you see here, acetoacetate. Okay, and here is the mutation. When there is a mutation in this enzyme, homogentisate dehydrogenase enzyme, that is lead to the alkaptonuria. Okay, so that is lead to the increased level of the homogentisate by the production or by the enzyme MAI, formal acetoacetate, and then to the fumarate acetoacetate. Also we have PBG synthetase enzyme causes like inhibition of the succinyl acetoacetate, that is causes Fanukai syndrome and blepharocarcis, and then the succinyl acetoacetate also lead to the production of the acetoacetate. If there is a problem here, that is lead to the hereditary tyrosinemia type 1. What's so important about all these things here, there is a, a mutation in the gene that is called this enzyme homogentisate 1,2 dioxygenase enzyme, and that is lead to the more accumulation on homogentic acid. So everything like black pigmentation of the eye, black urine, these are the symptoms that we can see here. There is a pregnancy test is called human chorionic gonadotrophin. It's not chronic, chorionic. That is related to the placenta and this stuff. So H is a human chorionic gonadotrophin, which is a pregnancy test. It's immunochromatographic test because we use antibodies. That's why we can say it's immuno and chromatographic test. The other thing that the molecular weight of a human chorionic gonadotrophin, it's about 38 kilodaltons. Heterodimer, that is composed of two subunits, alpha and beta subunits, present in the cytotrophoblast and syncytotrophoblast of the placenta. So two layers of the placenta present in the cytotrophoblast and syncytotrophoblast of the placenta. The first morning specimen are preferred for qualitative urine pregnancy because it concentrated of a human chorionic gonadotrophin. So if the woman want to test her pregnancy, we said to her, please test for the pregnancy in the early morning because the urine is really concentrated with a human chorionic gonadotrophin. Absent in a very early pregnancy or abnormal pregnancy. Human chorionic gonadotrophin appear in a test after the 7 days of the conception. So it will appear in a 7 days or after 7 days of the conception, can be detected in the urine after 12 to the 14 days. Actually, human chorionic gonadotrophin appears after 7 days in the blood. If we just make a blood test, it will show human chorionic gonadotrophin after 7 days of the test. If we want to make the urine, then we can do it after 10 to the 12 days. It's level lower than the blood. It's used only for the pregnancy. Why is the highest serum value of a human chorionic gonadotrophin obtained in the second or the third month of their pregnancy and then decreases after there? Why do you think the human chorionic gonadotrophin, you can see its level in the second in the second month and the third month, but after that it will be no human chorionic gonadotrophin because Now we understand why this, because it's an important question. Human chorionic gonadotrophin responsible for these functions. Human chorionic gonadotropins give signals to the ovaries to produce progesterone and estrogen. Human chorionic gonadotrophin ensures that the embryo has this necessary hormone for the growth and the development. Human chronic gonadotrophin modulate the mother immune response to the acceptance, the presence of embryo, and also promote the growth and the development of the placenta. But all this stuff important in the first 3 months, and you can see that. So the placenta develops shortly after the implantation. 6 to the 10 days of the placenta, or after the conception. But in 8 to the 12 weeks of the pregnancy start, the placenta takes the function of the human chorionic gonadotrophin. At that point, there is no importance to be the human chorionic gonadotrophin, so it start to decrease. So we want first, it's like in the first 3 months, and it's about, if we just said that is 3 months, that's mean it's 12 weeks. So from 8 to the 12 weeks, like from the first of the conception till the 12 weeks, the human chronic gonadotrophin mostly do all the functions of the placenta. It's important to produce the progesterone. Actually, it's just produces signals to the ovaries to produce more progesterone, more estrogen, and it also ensures that the embryo during the pregnancy takes enough food and for its development and its for growth, and also modulate the immune system of the mother. So it's modulate also the growth of the mother and her immune system, and it promote also the growth and the development of the placenta. So all these functions of a human chronic gonadotrophin, they are done in the first 3 months. But the placenta, yes, it forms after 6 days of the conception, after 6 to the 10 days of the conception, that is lead to the formation of the placenta, but it's not developed. So the human chronic gonadotrophin at that time make also the development and the growth of the placenta and do all the stuff like um, stimulate the production of estrogen and the progesterone, modulate the immune system, make sure that there is a development and the growth of the embryo. And then after the 8 to the 12 weeks, the placenta is old enough, its development is well developed, so it takes the function of the human chronic gonadotrophin. So the human chronic gonadotrophin has no action here. And we want to just discuss how it's happened here. As you can see here, it will, like the green one, alpha amylase enzyme, alpha amylase, and here is the human chronic gonadotrophin. This is the pregnancy test. As you can see, there is a control and there is a test, and here is the urine. Okay, so as you can see here about the control, immobilized antibodies against mouse antibodies. So here is antibodies also, and here's also antibodies. Okay, but these antibodies immobilized, they are just stick to the control area, and they are just against these antibodies to make them bind to them. What these red one, they are called conjugated monoclonal antibodies against alpha amylase enzyme. This red one that is present in the urine against alpha amylase enzyme, they are not against these antibodies. Okay, so the gold conjugated mononuclear antibodies against the alpha amylase enzyme, here in the test they are immobilized monoclonal antibodies but against a second epitope of the alpha amylase enzyme. Okay, so again, these antibodies, these are immobilized and these immobilized, but these against mouse antibodies and these against amylase. So the alpha amylase here bind to these immobilized monoclonal antibodies. These antibodies against the alpha amylase, so they are bind here and detect if there is a pregnancy or not. It's like a lateral flow strip test or immunochromatographic test, which is a pregnancy test, as we said, immunochromatographic test, because we use antibodies. And then we talk about the total protein period reaction. This test used to detect the proteins in the sample. So the total protein period reaction, it's actually the test that is used to detect the protein in the samples. The test performed in a basic alkaline solution. So the copper ions binds to the peptide bonds that is present in the proteins, and the copper make connection with the peptide bond and then give the blue violet color. The intensity of the color is proportional with the number of the peptide bonds. The color change measured by the spectrophotometric minimum measurement, the 3 amino acid. Because of 3 amino acids, so we have 2 peptide bonds. Okay, it's really the total protein period test. It's a important test that is used to detect the protein that is present in the sample. Actually, we put it in the alkaline solution. All the test that is put in alkaline solution and depend on the principle that the copper ions bind to the peptide bonds between the amino acid. And after the copper ions bind to the peptide bonds between the amino acid, so that is lead to the blue violet color. And if it increase the level of the peptide bonds, that increase the level of the blue violet color. That means increase the level of the peptide bonds and increase the intensity of the color. We can measure it by the spectrophotometer. And the total protein concentration that is present in the plasma, it's about the 64 to the 83 grams per liter. So that is just how is the normal protein that is present in the serum. It's about 64 to the 83 grams per liter. Radial immunodiffusion, I will explain in a very detailed form, but in a separate thing. The radial immunodiffusion, here we have the antigen, here we have the precipitate, and it will be antibody incorporated in the CAR. And again, I said this antigen diffusion that is present in the middle. Okay, but around this is a precipitate and form the ring. That's what we call them as a radial immunodiffusion. And as I said, it will explain in a separate video. It is replaced now because it's old one, so it's separate now and it's not used now. It replaced now, but it still has many advantages, like more simple, less expensive, minimal equipment, high sensitive, no special reagent that is needed for this reaction, and good for the routine testing. It replaced by the enzyme linked immunosorbent assay, which is known as ELISA. Enzyme linked because it associated with the enzyme immunosorbent assay, and we will discuss it later, and also we will discuss it as a detailed form. So it replaced by these two things, the flowmetry and ELISA. About the precipitation, if there is antibody excess, so all the antigen sites are covered. It just they separated into 3 zones: antibody excess, antigen excess, equivalence zone. Antibody excess, all the antigen sites are covered by antibodies. Lattice formation is inhibited. There is no lattice formation here. So in the case of the antibody axis, all the antigens covered by all the antibodies, the lattice formation is inhibited. Equivalence zone optimize or optimal proportion when 2 to the 3 antibodies for each antigen. So each antigen, it has 2 to the 3 antibodies, produces maximum lattice formation, maximum precipitation. Okay, so it will produce the maximum lattice formation. Here is the lattice formation because we have 2 to the 3 antibodies per one antigen. So that is lead to the lattice formation, maximum precipitation. And then the antigen axis, all the antibody sites are saturated. Triple, that's mean one antibody but 2 antigen, but there is no precipitate. As you can see here, the antibody axis and the antigen, no precipitate, no lattice formation, because all the antigen are covered by the antibodies, and here one antigen, one antibody and 2 antigens, so all antibodies saturated. But in the case of the equivalence one, there will be 2 to the 3 antibodies per antigen, lattice formation, maximum precipitation. About the structure of the antibodies, it composed of a constant region. Here we can say it's a heavy region because they are just constant, and then the light region. Here is the variable, and here the antigen binding sites. Actually, that's why we called it a variable, because this is the when the epitope of the antigen bind to these antibodies. The first one, they are glycoproteins. When I said the glycoproteins, that is composed of the glucose and the proteins, but mostly proteins. It's a Y-shaped protein, as you can see here. It composed of the 2 identical heavy chains and 2 identical light chains. So it composed of 2 heavy chains and 2 light chains. Light chains held together by the disulfide bond. See, we can see here the light chains. So we have 2 light chains, and these 2 light chains, they are held together by the disulfide bonds. The variable version of antibody determines the antigen specificity. So this variable determines the antigen specificity. To understand, there is something is called paratope and epitope. Epitope, that is present on the antigen. So we can say epitope of the antigen bind to the paratope of the antibody. So this green one that you can see here in the antibodies is called paratope. Okay, so it's known as a paratope, and the antigen that is comes from nowhere is called epitope. So when I hear epitope, that is referred to the antigen bind to the paratope of antibody. Here's just a comparison between monoclonal antibodies, polyclonal antibodies, and we have also another comparison between them, but it's okay to repeat things because repetition is good. Repetition, I mean. Monoclonal antibodies and polyclonal antibodies. Monoclonal antibodies are produced by single clone of the beta cells, and that's why we said monoclonal antibodies, because it is produced by the single clone of the beta cells. It's high specific to one epitope. It's really high specific because to the one epitope of antigen. Made in the lab by something is called hybridoma technology. So it's synthesis by hybridoma technology, and I will explain the hybridoma technology in more details in the separate file. It's highly pure uniform antibody population. It's used for the targeted therapy diagnosis research. It's more expensive and more stable over the time. But in a polyclonal, it's actually produces by polyclonal of the beta cells. It recognizes multiple epitopes, so it's not a specific, but it can actually recognize multiple epitopes of the same antigen, derived from immunizing animals collecting serum, so that it no need to the hybridoma technology. Mixture of the antibody less uniforms used in the assays, in the diagnosis and the research. It's less expensive and less stable. Production of the antibodies for a specific purpose. Why we do the production of antibodies. Inject the purified antigen posed by the second antigen injection. Take a blood sample, test for the reactivity, purify antibodies from the blood serum, and by this way we produce the antibodies. The first one that we just inject the purified antigen, and then we again inject another antigen because we just like post it, and that is lead to the reactivity to produce the antibodies. Then we take this blood antibodies. Several beta clones are reacting with the antigen, each producing a specific antibodies directed against various epitopes of the antigen, which is called polyclonal antibodies, which can be raised in rabbits, goats, and sheep. Monoclonal antibodies, actually here explain the hyperdioma technology, but I said that again, I will explain it in a separate file. Monoclonal antibodies, the antigen is injected to the mouse. So first we inject the antigen to the mouse, then we kill the mouse and take the spleen, and then from the spleen we isolate the beta cells and the myeloma cell line formation. Myeloma cell line formation is a tumor cell. So the first thing that we inject the antigen to the mouse will form antibodies. We kill the mouse and take the spleen, and then after we take the spleen we isolate the beta cell. Then there is a myeloma cell line, the myeloma cell line formation, who is diffused with the cancer cells, I mean the beta cells. So the beta cells fuses with the cancer cells, I mean here the myeloma cells, and that is lead to the immortalized beta cells. That's what we call them as a hybrid cells, because it composed of a beta cells and the tumor cells, which is called myeloma cells. And then we cultivate them in a one cell in a well dynasty, and these clones it will be expanded, and then we assay for the binding antigen, lead to form reactive and non-reactive antibodies. We just ignore the non-reactive antibodies and take the reactive antibodies. Again, this is a comparison between the polyclonal antibodies and the monoclonal antibodies. The polyclonal antibodies is inexpensive. Polyclonal antibodies is expensive. Inexpensive, expensive. The more expensive is monoclonal antibody. It needs a training actually, because it's not about just inject the antigen and take the antibodies. No, we have to deal with the mouse. We have to do the hyperdoma. We have to deal with the tumor cells. So the training is required. We use hyperdioma, take relatively long time to produce. If you think that hyperdioma technology it's easy one, no, it's not easy. Actually, it takes time. It's so expensive, and that's what is make monoclonal antibodies is not easy. So it will be generate a large amount of specific antibodies. It just recognize only one epitope on the antigen, and that what is makes it more specific. Once hybridoma is made, it's constant and renewable source, no or low patch to patch variability. In the case of the monoclonal antibodies, polyclonal antibodies, it's inexpensive. Skills required for the production is low, so like low skills required for the production of polyclonal antibodies is relatively quick to produce. It's easy, it's more quick. Generalized large amounts of non-specific antibodies, recognize multiple epitopes even in the same antigen. It has batch to batch variability. Then we talk about the ELISA, which is called enzyme linked immunosorbent assay, and also ELISA it will be explained in a detailed form. So we have the immobilized antibodies, as you can see here, and then this is labeled antigen. Okay, and then here we have the label antibodies and the immobilized antigen, and look what is happening here. So the presence of the antigen is tested. the presence of the antibody is tested. Immunoassay involve two formats: competitive and non-competitive. In the case of the competitive, the antibody is limited. Then more tested antigen, the less signal after washing. But in the case of the non-competitive, so the antibody is in excess amount, the more tested antigen binds, the stronger the signal. Okay, so it will be a large number of the antibodies. It's less sensitive to the variations of the reagents, so more robust method. So as you can see here, this is the signal intensity, and here the signal detection, and the response, which is the change of the energy, is directly with the change of the substrate. So about the signal detection, we have two types: enzyme conjugated and biotinylated. Okay, so the enzyme conjugated, we have antigen antibody, often an enzyme like alkaline phosphatase or horseradish peroxidase enzyme, or PNPP or OPD, all these enzymes. Okay, and they just form the color substrates to the colorless substrate to the color substrate by using this enzyme. One of them, maybe we use alkaline phosphatase, maybe we use horseradish peroxidase, or PNPP or POD. In the biotinylated, signals amplification through butene iodine enzyme. So this enzyme lead to the signal amplification, like making the signal more larger. So here, beta nitrophenyl phosphate by the phosphatase, so it colors compound by the base, it turns it yellow. And we have also O phenyl peroxidase, and then it turns to the yellow. And by this mechanism, but actually you can see that is little information about the ELISA. Actually we will explain more on the ELISA. I will explain also more in polyclonal and the monoclonal antibodies, and many topics that I mentioned, it will be separately discussed. Thank you so much for listening.